Well, thank you everyone for your time today. It's a pleasure to have people willing to hear scientists when we have some people, important people at the helm calling for not listening to science anymore. So I appreciate everybody's making the time to hear this. I try to, I will see how it goes in terms of speed, the level of questions that I get as I move through. But the goal of this lecture was to, number one, introduce myself. I'm Gerardo Morfini. I work at the University of Illinois, Chicago, in the Department of Anatomy and Cell Biology. And my interest has been for many, many years. I started being a person that started axonal transport. As I'm going to show you in the presentation here a little bit, my career took a little detour as I was learning that some molecular motors responsible for axonal transport were regulated by protein kinases, and that triggered what has been over the last 10 years significant work that we have done in a number of different neurodegenerative diseases and the role of protein kinases play in those diseases. So as a first slide, I just took myself the freedom to put what we have for many, many decades been the main source of studies for ALS. It came from patients affected by the disease, typically at late stages when biopsies can be obtained. And we learned through many years of research and observations that there's a dramatic atrophy of the spinal cord tissue, which is manifested here on the right slide. If you were to slice this spinal cord and look at the ventral horns of the spinal cord, you will see a massive degeneration of these large cells here, which are the motor neurons, which are absolutely critical for the execution of our voluntary movements. So for many years, a lot of the research was driven by what was the end state of the disease. But as the animal models became available, we learned a little bit more of what happened much earlier in the course of this disease. And this is what I consider a classic slide that came from the group of Jonathan Glass, which many of you are familiar with, that illustrates here sort of different sections here in the case of the ventral horn of the spinal cord, the axon, and the neuromuscular junctions at the very end of these axons. And you can see that in the so-called the most popular model of familiar ALS, the mutant SOD1 G93A model, this paper made a very good case that long before there is any disappearance or any loss of cell bodies, there's a degeneration and loss of connectivity with this neuromuscular junction. This is represented here at different stages. Even at pre-symptomatic ages, you already see some sort of disconnection, which is highlighted by neurofilament staining here. You can see that some of them don't quite arrive properly to the neuromuscular junction. This phenotype becomes exacerbated with time to the point that there's only remnants of these neuromuscular junctions as we enter as the disease onset proceeds, okay? So this is actually not that late in the stage of the disease. So what this left very clear, it was something that was already established for a number of years, but brought to an animal model with much more detailed study, was that ALS, the neurons that degenerate in ALS, degenerate with a pattern of degeneration that is known as dying back. Pattern of degeneration. And that some of the hallmark of that pattern of degeneration, which is represented by this cartoon here, is the loss of synaptic connections and the degeneration of axons, pathology of axons, long before any loss of the cell bodies. So here there's some, the red dots illustrate aggregation of the protein, which is a common feature of not just ALS, but multiple neurodegenerative disease. Neurons affected in those diseases seem to follow the similar pattern, although the type of neuronal cell type that is affected obviously varies according to the disease. But this is a very generalized mode of degeneration in neurodegenerative diseases. And the take-home message here is that we need to take, even though it's absolutely correct to try to promote the survival of these cells at late disease stages, I think that the possibility that we can intervene with the disease process, it gets better as we tackle what are the most early events in the course of the disease, such as the axonopathy and the synaptic pathology that I mentioned first. So that's the TAFE sort of has been a guiding principle in my case I know there's some people that went even earlier, tackled the disease from an early point of view, including the Cleveland lab here, which is to directly knock down the protein that is causing the problem. But for those of us who have been studying the disease as it proceeds or following closely what happens, I actually strongly believe that any potential therapeutic strategy that is aimed to slow the progression of the disease will have to have in consideration this loss of connectivity. We already have people who has succeeded at blocking cell death in ALS quite successfully, for example, by knocking down the executors of apoptotic components. However, this didn't actually translate into any form of recovery, again, emphasizing that it doesn't really help too much to have a neuron alive if it's not properly connected with its targets. So we need to do something to try to maintain this connectivity throughout the disease. And the question then comes is what makes motor neurons particularly vulnerable in this disease? And this is a slide that I really love and that I use especially for graduate students, because I didn't make this slide, but a colleague of mine whose name is Peter Hollenbeck, he actually drew a motor neuron at around the cervical level, not even the longest one that we have, and draw this cell body, which will be about 10 microns, 20 microns in diameter. And then decided to do roughly in scale what would be the length of this axon. And this axon here goes back and forward almost a few hundred times before it actually finishes in this neuromuscular junction. So I think that this is a really nice way to try to put a little bit more touch with the dramatic challenges that these neurons have in comparison to other neuronal cell types. And this is because this cell body that is located so distant from this synaptic terminal needs to provide all the materials that need to be trafficking through this axon before they reach the final destination. And this can be, like I said, in this case of this neuron particular, something along the order of 50 centimeters long. Now, in turn, this neuron somehow needs to have some information of what is going on at the end of this terminal. And for that, it relies on information that needs to return to the cell body to inform whether transcriptional changes need to take place, and even inform the cell if it's alive and performing properly. This is now well known that is due through trophic support from the target cells. But this system and the communication that goes between this cell body and the terminal is absolutely critical determinant of the survival and performance of this neuron. Now, a little bit, I just wanted to simply illustrate the complexity, because we often see cartoons. Cartoons are always very useful, but if we stick to them, we can be quite confused and oversimplify the problem. So things need to be transported. I know there's some evidence out there. It still is quite controversial about the level or the degree to which local protein synthesis in axons contribute to the axonal mass. But nevertheless, it's absolutely unequivocal. The overwhelming majority of materials that are needed to generate this axon and to maintain it throughout our entire life dramatically depends on the synthesis of proteins that take place in this cell body, which, as you may recall, is very, very far away from this terminal. So now these guys have a very unique challenge, which is that they need to deliver all what is needed for these terminals to be working properly. And that is due to a process that is known as axonal transport that, depend on the type of contents, can be classified in slow and fast axonal transport. The slow axonal transport is mainly related to the transport of cytoskeletal elements. It proceeds much, much slower than fast axonal transport. But just to give you an idea, for a neuron such as the one that we depicted before, for the materials that are synthesized to arrive there, it can take up to a day and a half, whereas the slow axonal transport, it might take over a year to reach the destination. So this flow is going on as we speak right now, and it's incredibly dynamic because we also have the material that goes back to replace the old constituents as they are used. Now, it would be a lot simpler if this will be sort of, to make an analogy, an electrical cable that is homogeneous throughout the entire length. But this is not the case. You have defined axonal subdomains of very unique protein composition. So I highlighted two that are some of the ones that we know the best, the nodes of Ranvier, which are illustrated here. So a vesicle, this is actually the distribution of sodium channels. Look how exquisitely discrete is this distribution, and here's the diagram of all these different protein components that are all labeled with different colors here that actually form this compartment and makes it specialized and make it functional. If you wouldn't have a big cluster of sodium channels located here, you wouldn't have saltatory conduction. Which this implies is that some vesicles, after their synthesis, those vesicles that are carrying those sodium channels, they need to be delivered here and not somewhere here. Okay, because in here they will serve no purpose. Conversely, some components of that are critical for synaptic transmission, such as synaptic vesicle precursors, they're not going to be delivered at this domain, but they need to be delivered in here. And if we're going to complicate things further, there's no one or two nodes of Ranvier, but there's hundreds, sometimes thousands of nodes of Ranvier throughout the length of this axon. So what is the implication of this? Is that there might be some exquisite mechanisms that allow the components that are being transported along the axon to be delivered at the right time and at their proper location. And I'm going to get back to this a little bit later, because these mechanisms are actually what take for this neuron to have all the components properly compartmentalized to perform specific functions. We know quite a bit about what the molecular motor proteins are that carry this transport back and forward. Today, by now, my former mentor, Dr. Scott Brady, was actually instrumental in discovering of the motor that carry things away from the cell body towards the axonal terminal. And that's the conventional kinesin, which is a motor protein, is a tetramer form of two heavy and two light chains that in turn interact with a number of different vesicles. There's complexity. This is an oversimplification. There's actually 3 types of these heavy chain motors and 2 types of these light chain motors that together can form a wide variety of combinatorials that make different flavors of these motors. And it is thought that this has to do with the selectivity of the delivery of different cargos that I referred to in the prior slide. Conversely, the motor protein that carries all the material that is old, all the neurotrophins that are actually endocytose, autophagosomes, and a number of different organelles is conveyed by this motor protein that is even more complex in composition than the kinesin, is known as cytoplasmic dynein, and it has a number of different subunits. I'm not going to go through all of them because they're quite complicated, as include the heavy chains, light chain, intermediate chains, and so on. So it suffices to say that there's a degree of complexity in these motors. They should not be looked at it like a single thing. So I'm going to get to Siobhan's favorite slide now, because what does this have to do with what I was talking about, the dying back pattern of degeneration that we refer to in ALS? Turns out that as the genetic studies became more and more widespread, there was unequivocal evidence that disrupting any of these subunits of these motor proteins results in degeneration of specific neuronal populations. And interestingly enough, in all those cases, these neurons follow exactly the dying back pattern of degeneration that I referred to in the beginning. Here's a few examples of those. This actually, I did not update this slide. There's now more mutations that have been found, but there's one in mutations in kinesin 1A now have been identified in association with degeneration of upper motor neuron. And there's a subset of these mutations that actually appear to be affecting both upper and lower motor neurons. And in fact, there's been proposal of calling some specific familial ALS variants on the basis of this mutant motor protein. For the cytoplasmic dynein, several of these subunits that you see here schematized in different colors, there's mutations that have been identified that also result in motor neuron degeneration. This is not to say that this is precisely ALS, but what the take-home message of this is that even small alterations in the composition and function of these motor proteins can result or suffice to promote degeneration of motor neurons. This was quite interesting because by the time that these mutations were coming up, already other people have observed alterations in axonal transport in the context of ALS. In fact, Don Cleveland was one of the first ones to report those, especially for slow axonal transport, although he hasn't done much for fast axonal transport, which refers to the transport of vesicles moving back and forth, which is a lot more prominent than the slow axonal transport and equally important. So this is an incomplete list of a wide variety of genes that have been found that when mutated result in the generation of motor neurons that lead to familial forms of ALS. And many of you are familiar with all of these proteins, so I'm not going to spend a lot of time. But it's actually nevertheless important to remember how puzzling is the fact that proteins that have no obvious structural or functional connection to each other, they still cause this type of degeneration for this neuronal population. What is it that is, how can we actually explain the extremely high variety of genes converging on the same phenotype? So there's some suggestions from many researchers that many of these may share some components in the pathogenic pathways that they trigger. And this is a little bit what I'm going to be talking today for the work that we have done. I have highlighted here 4 of the most popular genes that have been found in association with familial forms of ALS, and I'm going to be covering these guys through the talk very briefly. So knowing that there were deficits in axonal transport in ALS, and knowing that these deficits in axonal transport suffice to cause degeneration of motor neurons, a number of years ago when I was still a postdoc, much younger too, I decided to test whether any of these proteins actually have the ability to interfere with axonal transport in order to try to identify the mechanisms that underlie that axonal transport defect in the first place. But this is a challenging question because most of the systems that we have available today, whether we're referring to cell culture or to certain animal models, they rely on the expression of the protein of the cDNA, exogenous cDNA constructs that we usually introduce in them. They need to be synthesized in the cell body. So if we transfect a neuron with SOD1, that protein has to be synthesized in the cell body before it reaches the axonal compartment. What is the problem with that? That's actually what happened in reality. But if you want to understand effects that happen in the axon itself, or effects of, or if any of these proteins affects axonal transport, this is a problem because we now don't know if any problem that we observe here has to do with a problem that started in the cell body. All right, so this becomes a little bit the egg of the chicken kind of thing. Is it something that it really takes place autonomously in the cell, on the axons, or is the axonal degeneration a mere consequence of these of deficits in the cell body? That has been a long controversy that have not yet been fully resolved. So pretty much any of these models will actually have that caveat. But there is one exception, and this is my dear colleague, the squid oligopili, for which I travel about 1,500 miles every year from Chicago to Cape Cod to work with this guy for about 2 months when they're available and they're brought to the coast. They keep them in tanks, and we can actually use them for a very unique experiment that only very few people have access to, and that we wish more people would have access to. So I always asked, I had this technician of mine, Axon, for many years that I actually brought with me to the Cape in order to do these experiments. He actually loves the squid. And besides all the advantages that I'm going to mention in a second about this preparation that we do with them, I challenge everybody, unless you really like to eat mice, that you can do a lot more fun things with the squid after you complete the experiment than you will if you work with any rodent model. But that's fine. That's the part that we really enjoy when we go there. But just to give an idea of some of the incredible discoveries that have been done, include the discovery of the Axon potential by Hodgkin and Huxley, which were awarded the Nobel Prize. That was discovered in this preparation that I'm going to mention now. The motor proteins that carry anterograde axonal transport were also, conventional kinesin was discovered by my former mentor, Scott Brady, working at the MBL in this preparation. And a variety of different mechanisms underlying synaptic transmission were also discovered in this little guy here, because they also have what is called the giant synapse preparation that you can get from here. Here's an example or a schematic of what we do when we go to the marine biological laboratory in the summers. We take a squid, we can actually open the squid, and there's two large giant axons that run throughout the mantle here. They're so large that, in fact, my student here is actually holding one of those with two threads, okay? We're talking about 2-3 centimeters long axon, single axon, that can be actually removed with very little effort out of the body of this squid, then can be taken to a small dissecting table to do the fine dissection, and you end up with something that is pretty much the axon itself with a few glia around and the plasma membrane. And this is something that actually blows my students' mind when they go and do this experiment over there, because you can now take this guy and do what we call the extrusion. Extrusion, try to think of a sausage and the skin that is around sausage. Let's pretend that you exert the pressure here and you gradually pull out. And you would think, you know, my students say, oh, I thought it would be like water. That thing will actually just fall apart. No, it doesn't fall apart. It's actually a gel that keeps entirely sustained by itself. So that gives you an idea of the concentration of protein that we're talking about here, enough to keep the things hold it together. And I don't have time to show you that, but the ultrastructure of these is actually can be undistinguishable from an axon that still has the plasma membrane. Why is this important? Because now we have no plasma membrane and we can perfuse anything we want that normally we cannot do in any culture cell or in an animal model. The whatever protein, whether they are antibodies, peptides, anything you want can be perfused at the precisely known concentration. This is not something that I work a lot with culture cells and we put an inhibitor, and the only concentration we can be for 100% sure that we are dealing with is the one that we put in the media. But the amount that gets inside the cell oftentimes is not the same, okay? So bottom line is that we can now put things, expose these axons to a protein, an antibody, peptides, anything that normally does not permeate through that membrane. And we can do two types of experiment. We will do microscopic experiments where we can put this in a slide. Again, this is a naked axon just to show you the length. It's sitting on a cover slide. This is an Eppendorf tube to give you an idea. This 2 centimeters long single axon, we can actually put it in my special type of microscope and observe this movement that I was describing in the original slide. So what you're about to watch now is this is a schematic, a cartoon of, you know, represent the cell body, the synaptic terminal, and this material that will be moving in the video in this direction is so-called anterograde axonal transport. The one that comes back is called retrograde axonal transport. And this should actually impress you for the fact that it's real time. So this is not time average, like often you see these fluorescent movies where people capture numerous images, they play it very fast, and it gives you the false impression that this is happening at that speed. No, this that you're seeing here, it is 30 frames per second. This velocity, this type of trafficking is what's going on inside these axons. And immediately you will observe these sort of lines here that will actually be the microtubules through which these vesicles will be moving. And you will see some large vesicles right here, like these mitochondria. They're much slower. But there's a continuous stream of material that is moving in this direction anterogradely, and a lot of material moving in this direction retrogradely, okay? It's actually overwhelming. And you have to be thinking that you're only looking at a 200 nanometer section of this, when the preparation itself is about 0.5 millimeter thick. Okay, so it's absolutely remarkable the amount of vesicles that are moving here. What we can do with some fairly old but yet efficient equipment is to quantify this movement by having some cursors matching the speed of these vesicles, and then write it down. This is I put here a sort of an example of a typical experiment where you actually, each one of these dots it's a recorded speed that we took. And when we do many numerous repeats of this experiment, we end up with something like that we can integrate, and we can get the average velocity of vesicles moving in the anterograde direction and the average velocity of vesicles moving in the retrograde direction. Oh, sorry, I did not finish the video. Sorry, Shokan, I know this is your favorite, sorry. So I just was getting back to the cursor part. Can you guys see a little bit on the video?
Yes.
All right. Great. So these are the cursors that I'm going to show you now. They're going to come in a few minutes, in a few seconds. Once we got this, it's the longer version of the video. Just bear with me. This is also, we can actually go to the periphery and observe individual microtubules and see how things move along those microtubules as well, in case that we want to suspect that something is wrong. So, but the real part I want to show you is the cursors here. So you see these cursors that match, that you can actually make going in this direction to this direction. We have a number of observers. This is all published, so I'm not going to get in a whole lot of detail with this. But we finished with a lot of repeats, and when we integrate, we can conclude whether something had an effect or didn't have an effect on the movement of these vesicles. So what you see here is a classical control. And the velocities of the vesicles moving in the anterograde direction are slightly faster than the ones that come in the retrograde direction. And the reason for that is that the vesicles moving anterogradely are smaller in size than the retrograde. So basically there's a continuous flow of plasma membrane going up and down the axon. And if you disrupt that flow, somehow you're gonna have a deficiency, because all the membrane that needs to arrive needs to equal the one that comes back, or you will accept, unless an axon is undergoing active growth. In that case, there will be more material moving in the anterograde than the one that will be coming back. But in a steady state, mature neuron, this is actually quite balanced. So back to the question, sorry, I keep going on tangents here, but we got a hold of recombinant proteins, SOD1, a number of different mutations that we obtained purified from our collaborator. I want to highlight Dr. Brown and Daryl Bosco, very dear friends of mine. They're at UMass, and they provide us these proteins. And we now, because we can actually put this in the presence of the axons, we ask the question, do any of these proteins affect axonal transport? And a summary of that, which has been published a long time ago, is provided in this figure. You are familiar with this result already, because if you put the wild-type form of SOD1, you see that pretty much both anterograde and retrograde transport remain fairly unaffected. But when you actually introduce G93A mutant SOD1, one of the most widespread mutations that have been characterized in the ALS community, you see something very different. You now see a dramatic inhibition of anterograde axonal transport that does not compromise retrograde axonal transport. Is this something unique to this mutation? No, we found at least 4 more. I only show you 2 more here, G85R and H46R, and here's the quantitation of that. You can see that all these mutations that we tested, they will selectively producing this, qualitatively producing the same result. They would inhibit anterograde transport without affecting retrograde transport. This was important because it meant that whatever this SOD1 was doing, it didn't have anything with disrupting microtubules. Otherwise, we would have seen an effect on retrograde axonal transport as well. So there were a lot of hypotheses by the time that we got these results that have been put forward as to how this mutant SOD1 may be causing deficits in transport. And a favorite one, of course, for many people was that the aggregated protein somehow was capturing the motor proteins. So we did a lot of experiments, and we were lucky that another group in Japan were doing concurrent experiments, and we found no evidence of interaction between the SOD1 and any of the motor proteins, whether it's kinesin or dynein. So this is what is shown in here. You can effectively immunoprecipitate kinesin heavy chain. You see no traces of SOD1 and vice versa. So we found no evidence of that type of mechanism. But we were still, of course, being intrigued as to why or how does these mutant proteins interfere with this process. And one clue came from an experiment. This is the same paradigm that I showed you before, but in here we can take what we call sister axons. I don't know why we call them sister. It's probably because it's the Me Too movement. I like to call them sister axoplasms. So it's two axons that are extracted from the same squid, because these are wild creatures. So there's a little bit of inter-squid variability in a number of different things. So to make the perfect control, we take one axon, two axons from the same squid, and we can perfuse one axon with a wild-type protein, the other axon with a mutant SOD1 protein, and also spike this with some radioactive compound. In this case, we use radioactive P32, okay? So radiolabeled phosphate. And here's what you see when you now lyse these axons, you run them in a gel. That's what you see here, axon 1, axon 2. This is an antibody that recognizes neurofilaments, which are the most abundant proteins in the squid axoplasm. And these are the two bands that you see here. And now if you do an autoradiogram, a short and a long exposure, you can see very clearly that those axons that were perfused with G93A mutant, they display more phosphorylation of these neurofilament proteins than the sister axon that was perfused with wild type. So somehow this protein, and now we're talking about exclusively in the axons. This doesn't have to do with some non-specific effect in the cell body. These proteins can actually elicit activation of protein kinases in the preparation. And we went a little bit farther using antibodies against the motor proteins to see if they were also targeted by these kinases that are being activated. And we observed a significant increase in the phosphorylation of the motor proteins, both the heavy and the light chain subunits. So once we introduced the mutant protein, they were changing the phosphorylation of multiple substrates. And this is actually something that is one of the major hallmarks that everybody that works on ALS agrees on. One of the earlier signs of the disease has to do with aberrant proteins of patterns of protein phosphorylation and deregulation of protein kinases. So this is a very clear hallmark of any form of ALS. And in fact, many people use these neurofilament phosphorylation antibodies recognizing specific epitopes in neurofilaments as a measure of activation of protein kinases in the disease. For us, this was very important because I gave you a little bit of information in the introduction that by the time that I got these results, I have already published a lot of work showing that different protein kinases affect the activity of these motor proteins. So here's a number of them. I'm not going to have time to describe all and each, but what you need to remember is that each protein kinase affected a selected subunit of these motor proteins. So the net result is the same in the sense that they were all affecting axonal transport, but they do it through different means, okay? So, but we have built all this body of evidence with these guys act as switches that can actually turn off the activity of specific subunits and therefore the trafficking of these vesicles throughout the axon. So back to this experiment that I already explained, we're now using antibodies that recognize active kinases in the squid. We went on and we actually did a screening of several kinases that we knew were involved in axonal transport. And you can see, I'm going to bring your attention to this one here, the phospho-P38. This is an antibody that recognizes an active form of protein P38 MAP kinase. All right. Many kinases that we look, they don't seem to change between axon 1 and axon 2, ERK, GSK3. But when we did blood with phospho-P38, so we processed this axon, we observed that there was a dramatic increase in the activation of P38 in those axons that were exposed to the mutant protein compared to the sister axon that was perfused with a wild-type protein. Here's a number of them. I think the end of this was a 5. So this actually told us that somehow these proteins, these mutant SOD1 proteins can activate the P38 MAP kinases. But we still didn't know if this has anything to do with the effect on transport. To do that, what we actually did was something that we call co-perfusion experiments. So we now, besides perfusing the mutant protein, we also perfuse the mutant protein together with a pharmacological or peptide inhibitor of X or Y kinase. So if you recall, this is exactly the effect that you get with mutant SOD1 inhibition of anterograde axonal transport, that was not prevented by a peptide that actually outcompetes GSK3. So GSK3 was not mediating this effect. When we did it with an inhibitor of JNK kinases, we found no evidence that these will actually block in any of these effects. But the first surprise came when we tested an inhibitor of P38 MAP kinases, because that inhibitor significantly restored the anterograde axonal transport when it's co-perfused with it. You have to remember, this is the same one I showed you before, but with a inhibitor of P38. So somehow the effect is no longer visible. So this is telling us that that kinase, the activity of those kinase, that of the P38 kinase was needed to have the effect. However, because oftentimes inhibitors are as specific as the time that people use them, inversely specific, meaning that the more the people use an inhibitor, they found more other targets, right, off targets. So we were not satisfied with this. And we established a collaboration with Marty Watterson here in Northwestern that was working with some very, very high specificity P38 inhibitors that were completely different structurally to these particular inhibitors. And I'm going to talk about those towards the end of the talk. But this inhibitor is known as MW069 and now has been refined as MW151. It's a brain permeable P38 inhibitor. And you can see that again, this inhibitor completely blocked the effect. But if you take an analogous form of this inhibitor that is modified in a single covalent link in a way that is no longer makes it effective to be acting. So you basically have the same composition, the same formula, but organized slightly different. You don't prevent the effect. So this gave us significant confidence, together with the biochemical data, that this effect was mediated, the effect on axonal transport was mediated through P38s. Feel free to interrupt anytime. Now, many people, most people, including myself, when I was studying this, I will hear a lot about the JNK pathway, the P38 pathway, the ERK pathway, but less often you will hear about isoforms of P38, okay? And there's a number of isoforms of P38. In mammals, there's as many as 5, alpha, beta, gamma, delta, epsilon. And actually we, because we have the possibility to perfuse recombinant proteins in this preparation, we went on and perfused the 2 that are known to be expressed in neurons, which are the alpha and the beta. And you can see that perfusing the axoplasm with P38 alpha yields exactly the same result that we saw when we're putting the mutant SOD1, meaning that anterograde axonal transport was inhibited. But look how impressive, because P38 beta, which is a kinase that does a lot of the same substrates that P38 does, it has a slightly different effect. It actually inhibits both directions of transport, meaning that we should not be misled by the fact that they are isoforms of the same kinase. They still have somehow a specificity of their substrates, okay? So we were, after this experiment, we were pretty convinced that P38 has something to do with that. But how does it happen that P38 inhibits axonal transport? I'm going to speed up a little bit here, otherwise I won't get to the interesting part of the plan, which is that we found that P38, in fact, phosphorylate as these subunits of the conventional kinesin motor, and in doing so, it causes this protein, the phosphorylation happens at this interface between the head of this motor protein and the microtubules, and the consequence is that that motor protein no longer can walk along those microtubules. And I didn't have time to show you the data, but all of this is published. We tested this. Okay, this is all great, but this is a favorite question that I always hear through my grants. Is this just a squid? Why the heck do I care? So we obviously needed to move some of these studies forward to the mammalian system, and this is a little bit what I'm going to be showing you here. And we were, frankly speaking, quite impeded to do this, partially because of the lack of funding. But when we examine spinal cord tissue of naive, wild type, and mutant SOD1, we could confirm activation of P38 in the presymptomatic animals, the spinal cord. So this is a little bit what I was talking to you the other day, Shaban, about how we do the immunochemical analysis using this particular antibody. Other kinases were not affected by the, or not differentially affected between wild type and SOD1. And actually, if you use antibodies against specific phosphorylated epitopes in the spinal cord lysate, you see, for example, the SMI31 epitope, one that is very commonly used to visualize axons. There's no changes, no obvious changes. But if you use this SMI32 antibody, you see a dramatic decrease in the immunoreactivity. But this is a D-phospho antibody, meaning that these neurofilaments are heavily phosphorylated in the mutant mice, okay? So you see the inverse. When you interpret a D-phospho antibody, you have to interpret it backwards than when it's a phospho antibody. So that's what this result implies. So there's clearly an hyperphosphorylation of neurofilaments at specific epitopes in this tissue, and there's activation of P38 kinases. And we went on to look at...
Sorry, how far along into the disease was that? Like what age were these mice?
Yeah, I mentioned this is a day 60, so it's still considered presymptomatic. These guys will start failing at day 80 because there's a lot of G93A strains, and you should know that. But this was before you could see anything in the Rotarod test. And you can do the same by immunohistochemistry. At the glance, this is, you see in red is the phospho, the active P38, and then N staining to label neurons. There's obviously some level of activation of P38 here on these axons around this area. And this becomes very much more pronounced in the G93A SOD1 model. And if you do colocalization analysis, you see that indeed the motor neurons that are affecting the disease, they also have increased P38 activation. There's also some P38 activation outside the neurons that likely corresponds to glia, but we have not yet done all the co-staining to really make a point of that. And this I'm going to touch very quickly only because, and although there's so much done in this, and I don't have enough time today, but this begs the question of how does mutant SOD1 activate P38? This is unpublished and confidential data by now, but I will actually tell you that in order to give you this answer, you need to understand a little bit about the MAP kinase model and how these kinases work, because the MAP kinase is sort of a cascade that involves a MAP3 kinase, a MAP2 kinase, and the final MAP kinase. So this could be junk, this could be P38 or ERK, this could be MKK3, MKK6, MKK4, MKK7. The complexity is remarkable. But what we actually were able to look here, and all these interactions, these MAP3 to MAP kinase interactions are direct among these guys, and each one phosphorylate the other in a cascade. But using tools that allowed us to disrupt this interaction, we were able to map a subset of families of MAP kinases. I'm not going to have time to show you that. So if you put a peptide that is known as DVD peptide, and you block the interaction of the MAP3 kinase with the MAP2 kinase here, you can see that's the result here. You again have the toxic protein in the presence of this peptide is completely abolish the effect. If you use a different MAP3 kinase inhibitor, the CEP11004, you actually don't block that effect. So we think that we have mapped, at least partially, some of the MAP3 kinases upstream that this mutant SOD1 is activated. How does it activate this MAP3 kinase? It still is something that is we're working on, but we don't know. So this is a schematics that details roughly gives you a glimpse of what we found. We found that several forms of mutant SOD1 through a mechanism that involves MAP3 kinases, MAP2 kinases, ends up activating P38. And this P38 targets a number of different components. It targets the motor proteins. And as I mentioned before, by targeting this motor protein, it causes it not to be functional anymore. And it also targets neurofilaments, for which the consequences of that are not entirely clear, but might have to do with the axonal caliber, based on work like, again, from Don Cleveland, has done some pretty good work with that. And then we should not forget that in an intact cell, another thing that people working in ALS will pretty much agree on is that there are dramatic changes in the transcription of genes. And it turns out that P38 is a major kinase controlling the activity of multiple transcription factors. So I think that this is actually quite consistent activation of P38 with what we see in the disease. And others have shown significant activation of the in the sporadic ALS of this protein kinase. Okay, so is this all SOD1, or is something more relevant, okay, for other forms of ALS? I don't have time to show that today, but I should emphasize that we also show that not just mutant forms of SOD1, but also oxidized forms of SOD1 cause the same effect. So somehow this activation of p38 alpha is linked to misfolding of SOD1. And that was published, so I would refer you to the papers here. But we were interested also in this slide where I show you so many genes causing the same effect. And we decided to examine whether other mutant proteins that cause familiar forms of ALS may have a similar type of effect on axonal transport. The first one was FAS, and I won't have time to show you TDP43. You're gonna have to take my word for it. This is also unpublished. The FAS is published. And I'm gonna go very quickly because what we found was in an analogous manner as we observed with the SOD1 was that perfusion of the wild type form of the protein did not have any effect. You guys are already experts. This looked like a control that we showed before. But if you perfuse 3 different mutant forms of FAS, you observe a dramatic reduction in both anterograde and retrograde axonal transport. And significantly, once again, the p38 inhibitor was able to rescue that effect, all right? So I could talk for this entire paper, but I want to get to the point that I really got excited.
Right, so do you see this with when you express GP repeat?
We did not manage to do that yet, okay? So it's a pending experiment, but we assume, given the fact that they go through p38, that the active p38 will be doing exactly the same that we saw when we did the SOD1.
I see.
Did that answer your question?
Yeah, and I have an additional question if
yes.
So is this kinesin transporting specific vesicles, do you know, or?
That's a fantastic question, yeah. One limitation, which I should mention limitations of this model system, is that we cannot actually make any claims of what cargo is being moved that we quantify. It's a bulk of all the vesicles going there. So the question is, I suspect there is a subset of vesicles, but I will have to explain you why, and it will take me a little time, but I will tell you in 2 words that you remember how I mentioned that different combinatorials of these motor subunits create different motor flavors? Well, those different types of motors, which are defined by different composition, they seem to be associated with different cargos. So the fact that the p38, and now I'm going in detail, okay? The phosphorylation side of the p38 on the kinesin, it does not affect all the kinesin heavy chains. It's only 2 of them. There's Ki5A, Ki5B, Ki5C, and the p38 only targets Ki5B and C, all right? So it's implicit from there that only a subset of motor proteins is being affected by this pathway. Okay? All right, so, but I don't have an answer for your question in terms of which vesicles are actually being selectively affected here. But I do believe that it really doesn't matter too much in certain way, because everything that is sent over there is needed and is need to fulfill the function. So like I said before, it's like converges in axonal degeneration, but you can affect many elements of this transport and still get the same result, all right? A dysfunctional synapse and a dysfunctional axon. The sodium channels don't get clustered where they need to be clustered. Synaptic vesicles don't arrive where they need to arrive. They're all variations of a theme. So, and this, I only put this slide here because what it gets us very excited is that Marty Watterson has gone on to refine further the inhibitor that I showed you before was working so effectively. And this is a brain permeable inhibitor that has been shown unequivocally to work very well and very select. I will actually argue that this is the best kinase inhibitor ever done to this day. In terms of selectivity and efficacy, all right? And people has actually already shown very good properties of this inhibitor, not just in ALS, but also in mouse models of Alzheimer's and mouse models of traumatic brain injury, which all feature neuroinflammation and microglial activation with involvement of the P38 pathway, okay? So the neuroinflammatory component of those diseases is being actually targeted by this inhibitor very efficiently. However, the pathways that lead to the deficits in axonal transport, for example, in Alzheimer's and TBI and others, is completely different than the one in ALS, all right? So it's only, what we would like to do, and what actually currently doing with the help of NDR, is that we have acquired enough amount of this inhibitor to run a test in the G93 SOD1 model. But I'm gonna save all of that by February or March when this study is completed. So this brings me to the help from NDR and what we plan to do with some of the funds that they have provided us. We're really behind. We've been meaning to do this experiment for a really long time, which is what would happen. One way is to do it with pharmacological inhibitors, but of course that's very time consuming, and often certain labs such as mine were not suited to do that type of study with the number of personnel that we have available. But there's some other things that we can do in terms of proof of principle to try to determine whether reduction in the activity of this protein kinase has any ameliorative effect on any of these 1SOD1 and any of these ALS models. So life is complex and you have to pick and choose. We pick and choose an animal model that is very well established, which is the SOD1, G93A1 mouse model. And we propose experiments. This is taken from a grant that I was awarded recently from the NIH, where actually what we propose is to evaluate hallmarks of the disease and how they are impacted by reducing the activity of P38 alpha. So how can we actually in vivo reduce the activity of P38 alpha without compromising the mice themselves? And the answer to that lays from this mouse that was already acquired through NDR as part of this project. And I will disclose that we will be receiving pups hopefully in a month and a half, so establish this colony. These animals are known as P38 AF mice. AF is not where, I know in English people use AF for other things, but the reason for the AF has to do with two mutations that were introduced in these mice. So they are knock-in, meaning that one of the alleles encoding P38 alpha in these mice have been mutated in two sites that are important for the activation by the upstream MAP2 kinase. So they basically display 50% of the levels of the active kinase, and they have no phenotype whatsoever. They don't have any locomotor phenotype. They seem to, half of those of P38 alpha seems to suffice for some most basic functions. Of course, I take this with a grain of salt because maybe there's a challenge for which you need that. Nobody can say that any mouse don't have any phenotype. But I would say that as much as we can tell, we're not aware from the bibliography of any deficits that may complicate the studies that we want to do. So the goal here, what we are aiming to do, and I'm hoping to give you results 6 months down the line or 4 months down the line, will be that we're going to be crossing these animals with the G93A mice and a specific version of the G93A1 mouse that it features fluorescently labeled axons, and establish whether there's any impact on the axonopathy phenotype that we observed before, the neuromuscular denervation, demyelination, astrogliosis, or microglial activation. We have everything set up to do this. But you might say, well, how do you know that it's going to work? Well, what it actually allowed me to propose this experiment was that I was working on a parallel story that is not about ALS, but it's about Huntington's disease. And using the squid axoplasm, we were able to map a specific JNK isoform, another kinase related to P38 as a cousin, but that I already showed you data here that this ALS issue was not, deficits were not caused by JNK, but instead by P38. But in another project, the squid revealed that deficits in axonal transport caused by mutant Huntington were caused by a kinase that is known as JNK3. So we did in a study that is very analogous to this. We took these clues from the squid, these secrets that we extracted from the squid, and we went to an animal model of Huntington's disease that is known as R62 mice, okay? And we crossed those mice with YFP mice that express fluorescent proteins in a subset of neurons. And this is an example. So you produce now a YFP mouse reporter, and the brother or sister will be affected by the mutant Huntington gene. And these are examples of what you can see in the cerebral cortex or in the corpus callosum of these mice, okay? So we generate, this is what we call R62 reporter mice. So we went on to, because our objective was to establish whether JNK3 has anything to do with the degeneration of axons that happen in Huntington's disease model. So we created 3 lines of mice: the wild type naive, the R62 mice is in red, and in blue, the R62 mice that no longer has JNK3. And we did a number of locomotor assays. This is it's called the rotor rod. You put the mouse on a rod that is rotating, and you can quantify how efficient this mouse is to walk on that rotor rod. And you can see that, of course, the R62 performs very poorly in comparison to the wild type. But what it blew our minds was that actually deletion of JNK3 in this mouse model produces a very dramatic recovery on this particular phenotype. And that was true at 2 different ages. This is considered fairly late stage for the R62 mice. So actually, at least in this parameter was clearly recovered, and the survival of the mouse was much longer. And if you analyze the amount of synaptic terminals from the cortical neurons that arrive to the striatum, you can see that in the R62 there is a dramatic reduction of this bGLUT1 marker that is recovered again on the R62 mice that don't have JNK3. What about the axons? So here's an image of the corpus callosum, which is the largest bundle of axons that connect our 2 hemispheres in the brain. So you see a beautiful organization of axons in this YFP mouse reporter up to age P90, okay? So this remains constant in these mice, the organization of these axons in the wild type. But in the R62 mice, you see a dramatic degeneration of those axons. They don't look nearly as healthy as in the control. And if you now would section these axons in a way that the axons are coming towards you in the screen, is what you have in figure E. So this is a wild type. It's all these yellow dots are axons that are running towards you in the corpus callosum, and the oligodendrocytes that are over there for the most part. There's some glia as well, but those are staining with the blue DAPI. There's a massive degeneration of those axons at late stages in the R62 mice, but deletion of the JNK3 gene significantly recovers that. So what we're hoping is that by using the same principle, this is actually images that we took from the spinal cord of G93 SOD1 mouse with a YFP reporter, so we don't even need to stain this. We just simply section the spinal cords and we can see the axons there. This is the beautiful organization of axons that you see in sagittal sections of this particular YFP reporter, the neuromuscular junction. And then you actually can see a dramatic degenerations of those axons in the G93A mice. So what we hope is that what we would like to have some months from here is the images that will correspond to the same mouse here, but that features an attenuation of the P38 signaling. If that works, it will be a very nice proof of principle, because genetically we're removing a specific molecular component that will encourage us to move forward the pharmacological experiments with the MW150 inhibitor that we have access to. So with that, I will only mention that all this work, there's a lot of people here that were involved in this, but the main ones that I want to mention are my former mentor, Scott Brady, with whom I still go to the MBL. This was the first year in 20 years that I didn't go, which broke my heart. And Yu Song, which is another colleague that works at Harvard University that comes over on the summers and spend time with us, and Daryl Bosco and Dr. Brown at the University of Massachusetts. So with that, I'm happy to take any questions, but I hope that I was able to explain what my interests are and what I'm working on, and what, you know, the foundation here will actually is helping us achieve.
Does anybody have any questions?
I have another question. I don't know, is
Which one is
maybe,
more?
so in this squid axon, did you try to exacerbate the anterograde transport for longer time? Because I think it was minutes, right? That after you infuse the protein?
Well, no, actually, we cannot. We really, so the squid is not amenable to, let's say, overexpression or transfection or anything of that sort. So we cannot afford that experiment that you're suggesting. But I do think that transport itself is not going to be a good way to correct the problem, because the kinase will still remain active and will still be targeting those motors. That's why, even though I'm axonic transport, I've always said that I'm a closeted kinase guy. because actually I use transport as a readout, but what really I'm interested in there is to see how these proteins activate kinases that are in the axonal compartment. So your point is well taken, but we will have to have a lot of knowledge about what you brought up early, which cargos, before we could actually make a guess on there. Whereas by tackling the kinase, which is upstream of the transport problem, I think that our chances to really make a difference are higher.
Yeah, indeed, I was wondering if you, I don't know if it's possible in the setting that you have, or exacerbating a little bit longer, like the anterograde transport, if you could then take the axoplasm and check by mass spec also the protein differences that
that's very, you read N2 of that grant that I put there.
No.
No, it's a very good question. In fact, one of the things that there's been challenges because it took really long time to get genomic data from the squid. And I'll tell you why, and this might blow your mind like it did when I heard the first time, but it turns out that the squid genome is about 11 times higher in size than ours. And to that, you still need to add RNA editing. These guys are masters of RNA editing. So the complexity of the genome was fairly high until people that are specifically doing that were able to start pulling out sequences that we can work with. So this next summer, if things go well, we have a fairly large now database where we're actually going to be doing the type of experiments that you suggest. More specifically, what was proposed was to perfuse, of course, with wild type and SOD1 and examine phosphopeptides using TNT labeling and mass spectrometry. If we get any interesting axonal candidates, the relevance of what you're mentioning here for the rest of the group is obviously that we may have biomarkers that go in association with P38 activation. So in the future, we could look at those biomarkers and infer from there, oh, in this guy, there's still activation of P38. So that's a very good question, and was actually the second aim, trying to identify what the downstream targets of P38. We already know that the motor proteins are and that the neurofilaments are, but I suspect that there will be no kinase is meant to phosphorylate a few substrates. They usually phosphorylate a large number of substrates. So that's a very good question. We still don't know.
And also you may discover which are the specific cargos, maybe some hints of which are the specific cargos that are affected.
It could be. We have to see how it goes. These are all very pilot. The preparation is fairly big. I'm actually going to go 2 weeks from now to MBL just to dissect a few squid that I can bring and kind of do the proof of principle. So the first experiment will involve simply axons with and without. I will phosphorylate, add extra P38 to an aliquot to see if the method allows to see differences. If it does, so we have scaled up the experiment to really do what you're talking about, but it will take work. Because as you know, there's a lot of false positives and false variants. So we want to make sure that we do it maybe slightly slower, but surely that our method can actually distinguish these alterations. Otherwise, you end up doing what a lot of these mass spec studies do, which is the cherry pick your favorite one.
So Gerardo, just very nice talk. I really enjoyed it. Yeah, I don't do that much in the ALS world, but I'm getting more involved, as you can tell.
And I follow your stuff in Parkinson's, so I know there's analogy there. It's another dying back.
Well, yeah, that's something I was going to come to, is around specificity. The P38 it's a very nice story, but it fits into most cell types, not just obviously neurons, but also glia and astrocytes, and it fits into most diseases as well. And one of the things that I'm always coming back to is specificity and precision in this. And
Yes.
And one of the things I was going to point out is that I'm really interested in axonal dying back and thinking about how that occurs and why it's occurring, and what is the molecular machinery underlying it. And I'm thinking of this generally in the context of PD and generally in dying back axons in striatum, and then the fiber tract back to the nigra. But the point I was going to make was around dynactin and P150 glued, because there you've got this absolute specificity. You've got amino acid changes within 10 amino acids, from D59S in the familial motor neuron disease case to G71R, for example, 12 amino acids difference, where you have motor neuron disease in one and completely spared nigral neurons, and in the opposite case you have loss of nigral neurons but completely spared motor neurons. And yet it's both.
You're talking about like Perry syndrome, right? Perry syndrome.
Perry, yeah, and they're both TDP-43 proteinopathies, and so mechanistically there's something very, very specific and selective about it.
Yes.
And I'd love to get a handle on it. So I'm planting a seed there to because I'd love to talk to you about some of that.
Yeah, we will.
Down the road, and some of the experiments we're doing, and perhaps get some insight from you, and perhaps get some calamari too.
Well, that will be good. That will have to wait, but I always invite everybody to come, and people love to stay there, see how we do the axons.
Right.
So you're more than welcome to come. Now, just only one comment about what you're saying. It's actually quite fascinating what you're talking about, where the mutations will cluster, having such a dramatical differences in the cell type affected. So one thing I like about the story, and I did not have time to show you all what we have done in synuclein because it's unpublished, but we have done all these experiments in synuclein, and we have mapped protein kinases that are actually critical for the toxicity of synuclein in this cellular process, and I'm happy to share that with you in another moment. But a lot of a major problem that has been with this is that these cartoons that I refer to often create expectations on the scientists that actually are very hard to confirm later on. Let me give you an example with P150 glued, and I'm a motor guy, okay? So a lot of people have actually talk about P150 glue. You will see all these cartoons. It's always in front of dining, working like a blind guy, touching the micro. There's no evidence whatsoever that makes clear what the P150 glue is doing. Moreover, P150 glue is only linked to a tiny fraction of the cytoplasmic dying. And we know that from metabolic labeling experiments that were done a long time ago, where you can follow how the different proteins were trafficking. And they don't traffic together in the complex that you constantly see in all those cartoons.
Right.
So what I suspect is the cytoplasmic, that particular subunit of the dynactin complex has other functions that are related to dyning, but not necessarily linked on this view that is now becoming sort of a dogma, how people draws them together, okay? So it's a little bit of, sometimes we have to re-educate and question that, where does that scaffolding or that mega Dalton complex that you're doing there, where does it come from? And when you start looking really carefully, you often find that these complexes don't resist the most minimal analysis that will allow you to make an inference that they do exist as a complex, okay?
I think that's well said. I blame Erica Holtzbar.
Oh God! Well, you're very direct. I have to be very polite, but I will keep quiet.
I
will not. But yeah, they were very misleading. There were a lot of. In fact, okay. So these vesicles have all those vesicles that we see moving. They have a diameter between 50 and 80 nanometers. people will draw Huntington, which is the 3,000 amino acid protein. The whole dynactic complex, which has how many subunits with the ARBs and all stuff, on its own has 8 subunits. The whole dining complex, which is another 1.3 million Dalton, okay? And all of that thing and the kinesins with so much comfort. If you would put all those proteins together, they are larger than the vesicles themselves.
Yeah, sure. Yeah.
And when we do, and if we look at the cryo-EM work, which was done back in the days when people were, you know, they didn't have the methods we have now, but they were careful about the things. And look, you see that the link that links the vesicle to the microtubule is no more than 10, 20 nanometers in length. So, but like I said, people don't read the old literature anymore.
No, except the old ones. We read it as they came along, yeah. But yeah, no, I agree. Yeah, interesting. Anyway, it'd be nice to catch up sometime. I'd like to.
We will. I would love to talk more. So yeah, and I think you will enjoy the Parkinson story because it's actually more dining related than it is kinesin related.
I'm more moving into lipid biology.
I see.
So phospholipases. Yeah, but with your synuclein work, you might want to. We should talk about that.
But we'll chat. Definitely, we'll chat about that. Yeah.
Any
other questions?
Anybody else?
Yeah.
Anybody else have any questions? No? No? I guess we're all done.
We lost David, no?
No, Steer, do you have any questions? David,
no, I mean, that was a fascinating talk. For somebody that's not an expert in the field, it was just absolutely stunning, I thought.
Well,
You did a
thank you
great
so
job.
much. I appreciate that. Very nice.
It's the Axon movie, isn't it? That's what got you. It's those moving axons. Anyway, thank you very much. I did record it. I will edit out just the beginning part, just leave you with your PowerPoint if I can. And hopefully when I lost my earbuds, we didn't lose the sound on the presentation. And then anybody that wants the presentation, I will have them email you asking for it, and I can put it in a Dropbox.
Yeah. I'm
I'll
happy
put it in a
to
Dropbox for you.
We love to share. You know, this is a nice group of people. I'm not concerned about that. So just want to I'm only extra careful sometimes because some of this work was done with other people and I cannot talk for the others. But for me, I'm not so concerned. We have a lot of ideas. We just don't have sometimes the means to test those ideas. That's what limitation is.
Okay, well, sounds great. Thank you, everybody, for coming.
Thank you, everyone, so much.
Thank you.
Gerardo,
Look forward to the next one.
thank you, Gerardo. Great presentation.
I'm
And
glad
then
you enjoyed
help
it.
you with the means.
And yeah, you guys already are doing it. Yeah.
Yeah.
Thanks so much.
Challenge us to do more.
All right. Well, we'll be talking about that.
Okay,
Have a good
thanks.
one, everyone.